So our first speaker today is Rebecca Metivier, who is here with me in my office. So Rebecca received her BS in marine biology from Roger Williams University and her MS in chemical biology from Boston College. After receiving her master's, Rebecca worked as a research associate in the proteomics group in the Center for Protein Degradation here, where she was very focused on target identification using established methodology, as well as leading the development of new workflows within the group. From there, Rebecca then joined the TPD Proteomics Core as a scientist in 2001, where she's really gone from strength to strength in terms of developing methods here in the group. And at the moment, she is now the TPD Proteomics Core Director, where again, she's still developing a lot of methodology for screening degrader molecules and really expanding on a lot of our efforts, looking at degraders in induced proximity. So I will pass this over to Rebecca to tell us about some of the work she's been doing recently. Bad timing. Sharing the correct screen. Awesome. Great.
Hello, thank you for joining today. So I'm going to be talking about some work that we've been doing in our group using proteomics to kind of unveil these interactomes of ligase and compound combinations. So I'm sure most in this crowd are now familiar with the process of targeted protein degradation. So just to briefly summarize, TPD involves hijacking the cellular protein degradation system by linking a novel target to an E3 ligase using a PROTAC or molecular glue, which then leads to the subsequent degradation of this target protein. This can be highly beneficial in diseases such as cancer, where we can rid the cells of disease-promoting proteins. There are several methods that are currently employed to study this protein engagement and subsequent degradation following treatment with a PROTAC or molecular glue. In the lab, biochemical methods such as TR-FET screening and Western blots can be used to measure protein binding, ternary complex formation, and protein degradation. Further down the line, phenotypic screens like cellular death or proliferation assays are used to quantify treatment effects. And also with the recent expanse of AI and protein structure predictions, computational methods have emerged to predict how proteins and compounds will interact. Even when used in combination, these methods cannot give a complete picture of the effects and off-target effects of these drugs. In addition, several require preliminary existing knowledge about the expected targets of these compounds. Thus, we come in. Proteomics can give a more complete picture of what's happening within the cell following degrader treatment, including any off-targets and insights into target specificity. Proteomics is currently used in several different ways within the field, from whole cell degradation proteomics, compound binding analysis, and measuring downstream protein activity following this compound treatment. In some ways, however, current proteomics methods can still be quite limiting, as they can't answer all of the possible questions surrounding a degrader molecule. Some of these existing questions include, what about proteins that are engaged but not degraded? What about protein family members that may not be directly bound or degraded but are still affected by the molecules? And also in conjunction with that, could these engagement effects still have therapeutic benefits? Also, how do we study compounds that have promise but aren't fully optimized for their intended effect yet? For example, they may only promote weak degradation that cannot really be detected, but they bind very strongly to their targets. So we want to know, is there a way to answer these questions? One way our group has started to try to answer these questions is by enriching and studying engaged targets through immunoprecipitation. Through this technique, we can capture the ternary complex before the protein would have been sent for degradation, which allows us to ID proteins that are linked to a ligase through a molecular glue or PROTAC, even if they would not eventually get sent for degradation. When comparing conditions to a vehicle control, we can ID newly engaged targets as well as off targets and other family or complex members that may be piggybacking on these direct targets. There are a few immunoprecipitation methods that our lab has used previously. Endogenous enrichment of a ligase of interest involves in-cell treatment of compounds and a pull-down done by a ligase-specific antibody bound to something like protein AG beads. This technique, while viable because it retains cellular context and endogenous protein levels, can cause difficulties if the ligase antibody does not produce a strong enrichment or if the endogenous levels are too low to detect. Another option is transfection of tagged proteins into cells, followed by pull down on the tag that we add. In this example, it's flag. This is another method that allows for retention of cellular context as the treatment is happening in cells. However, you are limited to cells that can be transfected, such as HEC293 T cells. And one could argue that the overexpression of the flag-tagged ligase is not a true physiological condition. So you might be pushing your cells into stress state. And then lastly, a tagged protein expressing stable line can be generated. This also allows for easier pull down using ideal tags such as flag. And it retains cellular conditions with the treatment happening in cells. But this cell line generation is very time consuming and not all cell lines are amenable to it. So you might not be able to use relevant cell lines to study. Each of these techniques has its own set of pros and cons, and our group actually decided to pursue an in-lysate immunoprecipitation method to give us more control over the conditions that are being used. Our lysate IP method allows us to screen compounds more efficiently and in broader contexts. We combine cellular lysate, a recombinant tagged ligase, and the compound in a tube. This allows the ternary complex to form in vitro, which can then be enriched on the conjugated beads. The in-lysate method allows for the use of virtually any cell lysate, not just cell lines amenable to transfection or stable line generation. So this gives us more freedom in what cell lines we can use. The tagged protein is also artificially spiked in without the need for transfection or the use of endogenous antibodies. We also mostly use flag tag protein, which allows for cleaner and more consistent pull downs over other tags. Besides its many benefits, this technique does have at least one major downside. By conducting the assay in lysate, we are losing cellular context and physiological conditions, which means we may be pulling down proteins that would never be engaged in a true environment. Thus, we generally independently validate any new hits that we get from this assay. We can use this immunoprecipitation proteomics platform to achieve two goals. The first is to identify new targets that might be amenable to degradation within existing chemical space. By measuring targets that are engaged, but maybe not degraded, we can identify new proteins that are able to form ternary structures with our ligase, indicating that they might be amenable to degradation with the right conditions. The other way we can use this platform is to screen previously untested compounds and chemical fragments to discover modalities that have the potential to become degrader molecules. If these compounds were solely screened in whole cell degradation proteomics, for example, they might be dismissed as dead compounds with no degrader activity, even though they do bind to targets. In initial pilot studies with this method, we were excited to see that we did indeed capture many more targets that are being engaged by, in this case, pomalidomide than we saw being degraded, backing our hypothesis that degradation proteomics is not necessarily capturing the whole picture. At this point, we were convinced we had a powerful TPD tool and we wanted to use it further. So with this new capability established, we set out to screen 20 existing cereblon binding IMiD derivatives in 2 different cell lines. Each of these compounds had previously gone through our degradation proteomics platform, but we now wanted to resolve their interactomes. In doing so, we were able to tease out a total of 270 previously unreported proteins being engaged by these compounds. This is just one example of a degradation plot versus the pull-down plot that we produced. And this interactome of FPFT2216 really stood out to us, as the interactome was really quite more expansive than the degradome of the compound. And so we decided to follow up on some of these newer or interesting hits that came out of this screen. In the example of this compound, we were looking into PPIL4, which was not seen as being degraded based on these filtering conditions. After we confirmed the target through biochemical methods, we initiated a TR-FRET compound screen to find a PPIL4 binder that was stronger than the FPFT compound. Feeding the top hit from that screen, which is that red dot we refer to as Z28, back into our IP method, we were able to see that it was a much more selective PPIL4 engager than the FPFT compound. Z28 also shows promise as a better PPIL4 degrader than FPFT, but some still work is being done in that front. So this is just one example of how we can use IP screening to identify and improve upon new targets and new compounds. Another important piece of information exposed by this initial screen is that the use of different cell lines can be essential in revealing a more complete interactome of the compound ligase pair. Looking at the targets of pomalidomide in MOLT4 and Kelly cells, we see quite distinct interactomes. And so we're seeing targets that would be missed without the use of this cross-cell line screening. So this brings us to the need for high-throughput proteomics. Due to the flexibility of this platform, the need for cross-cell line screening to reveal different targets, the availability of E3 ligases that there are to test, and the extensive chemical space within the TPD field, we can really expand our understanding of compound-induced interactomes immensely, and then use this information to develop new degrader molecules that target new therapeutically relevant proteins. So our group set out to adapt our current IP protocol to have high throughput capabilities. The IP platform had three major aspects that we wanted to optimize in order to successfully adapt it to the high throughput screening. And these were improving sample-to-sample consistency, the length of time it takes to prep the samples, and the time these samples take to run on the mass spec. So the first part of this process that we started to optimize is sample consistency and sample prep time. If we really want to have successful high throughput screening, we need to be able to produce extremely high quality data in a timely manner. So to accomplish this, we turn to robotic liquid handlers. The first liquid handler we used was an Opentrons OT2 robot. So with this, we were able to achieve moderately consistent samples, and we can prep up to 96 samples at one time. And with our standard practice of prepping conditions with 4 replicates, that means we can test up to 36 conditions per week. While a great tool in the lab, this robot is quite small as it's a benchtop robot, and it also requires a lot of human intervention during the sample prep, which ultimately defeats the purpose of using a robot. So we looked to improve on our automated prep and decided to build our protocol on a Hamilton star. So this robot is much larger. It has the capability to prep 2 plates of samples at once and in a shorter time frame than on the OT2, and also importantly without the need for human intervention. Also, it promises to deliver even more sample-to-sample consistency. So with our sample throughput now greatly increased, we now had a disconnect between the number of samples that we could prep per week and the number of samples that we were able to run on our instrument. With our current instrument setup, we can only run 224 samples per week, and that is assuming full run capacity with no other samples to run except these IP screens, and that also means no instrument issues, which is almost impossible to avoid, unfortunately. So we really needed to look at improving how long these take to run on the mass spec, so we could increase our throughput on that end. So we decided we needed to find a way to shorten our sample collection time without losing important data from the samples. With our current setup, as you can see, we can test 56 conditions per week. So we decided to invest in EvoSep1 LC, which allows for much shorter sample gradients and can run up to 500 samples per day. Well, we wanted to minimize the sample time but maximize information retention. So we set our sights on the 200 sample per day method, which would allow us to test up to 350 conditions per week. The big question that remains is, are we actually able to limit data loss with this shortened gradient? We compared a sample of our favorite test compound, pomalidomide, on both LCs. And while some lower abundant hits were lost on the EvoSep, we believe that the speed of the data we were able to collect outweighs this loss. Another compound we tested, SB1G187, is a promiscuous kinase degrader and engages many more hits than pomalidomide. So due to the increased number of targets, the data loss was a little more significant on the EvoSep for this molecule. However, we do rationalize that any compound that has a profile like the one on the right from the EvoSep would still be worth further study through traditional low throughput methods to confirm any of those hits. So we still see the value of the EvoSep as a high throughput screening tool. With the EvoSep, we can increase our data collection speed and have minimal information loss in the process. And also, interestingly, another benefit of the EvoSep is that there's no pre-run sample desalting step required, which in turn makes the sample prep easier and faster. So this increases the theoretical number of plates and conditions that can be prepped on the Hamilton robot. So with these newly optimized conditions, we have shifted the balance in the right direction, where we can run more samples on the mass spec and we can prep on the robot, even with the increased theoretical capacity on the Hamilton with no desalting step required. Most importantly for our group, this still leaves us room to run other non-high throughput samples on the same instrument. I hope during this talk I was able to showcase the power of our immunoprecipitation proteomics platform and how it can use high throughput screening to expand knowledge around degradable targets and degrader molecules. And we just really want to get our hands on some compounds and start testing them. So I would like to thank all the people along the way that contributed.